Welcome back, everybody. We're ready to start this next session. It's called Behavioral and Biological Convergence. And the driving questions are that we're interested in the connections and discontinuities between brain activity and behavior, and between psychological health and brain health. What can behavior and life experience suggest about brain health and resilience? So these talks are going to touch on some of the underpinnings of behavior in the context of cognition, emotion, and psychiatric disorders. Our first speaker is Dr. Monica Rosenberg from the University of Chicago, and she'll be talking on the neural correlates of attention and cognition.
Great. Thank you so much for inviting me. Can everyone hear me okay? Okay, perfect. So I'm excited to talk today about how we might characterize attentional and cognitive processes with predictive models based on brain data. And so when I was thinking about the concept of brain health, I think sort of generally this involves our ability to safely and successfully navigate the world around us. And when I think about how to do that, one big process that we rely on in order to, execute functions in our daily lives is attention. So just as an example that many of us may have experienced, we might be driving for a long time and sort of space out and miss our exit, right? And this is a common occurrence, but it can have sort of really negative consequences. So an article a couple of years ago pointed out one potentially negative consequence of an attention lapse in this context, and that is sort of a spike in car accidents related to people being distracted by what's going on their phones. Right, so attention is critical not just in this adult period as we're driving, but it is important for life outcomes across development. So for example, in childhood, we know that children who pay better attention have better educational outcomes during their schooling years. And so people differ in their ability to pay attention, and attention also changes over time. But how do we measure these differences between people and differences within people? This is just a subset of the psychological tasks and questionnaires and clinical measures in the literature. You might see your favorite task on there, and if you don't, please email me, because I'm always trying to add to this list so it's as exhaustive as possible. But what I want to propose in my talk today is that we can actually use brain measures in order to complement these behavioral measures of attention. And so in particular, I'm going to be mostly focusing on one type of brain measure, for 2/3 of my talk, and that's functional connectivity. And so just to be specific about what I mean by functional brain connectivity, when we measure functional connectivity, we scan someone's brain in an MRI scanner, and we divide the activity up into several hundred regions or nodes. And then we look at the activity signal time course in one node, and correlate it with the signal time course in every other node, which generates this, what we call whole brain functional connectivity matrix, or connectome. And so here, rows and columns represent distinct brain regions, and cells represents the correlation between activity in those regions. And so we can project this matrix back onto the brain like this. But I like this figure because it reminds me to emphasize that those lines you see are statistical interactions. So they're correlation coefficients. They're not necessarily structural connections between brain regions. So why might we think that functional connectivity would have any relevance to differences in attention between people or changes in attention over time? Work from Damien Fair's group and from my colleague Emily Finn suggests that every person has a unique pattern of functional brain connectivity, or a functional connectivity fingerprint, that's relatively stable over time and contains information about cognitive abilities, including fluid intelligence. So that's why we're focusing on this measure. And so as I'm talking about ways to predict attention and other abilities, I sort of want to argue more broadly that these kinds of predictive modeling approaches can help us move fMRI from a science of group averages, which is extremely important work telling us what happens in the brain on average when people pay attention, but it can help us move in the direction of moving fMRI towards a science of individual differences, right? So what can we learn about an individual person, their abilities, their outcomes, what treatments might potentially be best suited for them from their single brain scan. And so just as a brief outline of my talk, I'm going to be arguing that we can use brain-based models to predict attentional abilities, to capture changes in attention over time, and then we can use a different measure of brain activity to characterize individual differences in working memory in development. And so I want to start by talking about our work predicting differences in sustained attention in adulthood. And so in order to do this, we brought 25 adults into the lab. This was data collected when I was a graduate student. So this was a lab at Yale. We had 25 healthy adults come in and do a challenging sustained attention task. And I'm going to give you a little taste of that task now. Okay, so you're going to see pictures of city scenes and mountain scenes. And your job is to clap every time you see a city and not to clap when you see a mountain. Okay, clap for cities, not for mountains. They go by pretty quick, so just do your best. All right, here we go. So I like this task because even in really high performing groups of people, so like this group here, we still see some variability in performance. This is a challenging task which you did for about 10 seconds, but we had participants do the task for about 40 minutes in the scanner. They did get some breaks, but it was certainly challenging even in our healthy young adult population. And so the way we're operationalizing individual differences in sustained attention is with performance on that continuous performance task. So now for each of our 25 adults, we have a measure of whole brain functional connectivity collected as they were performing the task. And then we also have their task performance. So here we're using D primer sensitivity, which is just participants' hit rate relative to their false alarm rate. And the goal of this project was to use the brain data, so the functional connectivity patterns, to predict task performance. And in order to do that, we used a method called connectome-based predictive modeling. And I'll walk through it relatively quickly here, but I'm happy to answer questions if people are interested in the details. I will point out that all of the code associated with this technique, visualization tools, and a detailed protocol are available online. So if you are interested in applying the technique to your own data, I encourage you to check that out. The first step of connectome-based predictive modeling is to identify which functional connections in our whole brain pattern. There are over 35,000 in this pattern, so we want to know which of those are relevant to our behavioral measure of interest. And so to do that, we leave out data from a single subject, and now we simply correlate the strength of every connection with behavior across the remaining subjects. So that generates this matrix here, which just indicates to us how the relationship between a functional connection strength and behavior across individuals. And now we simply retain the connections that are most strongly related to behavior in the positive and the negative directions. So the network on top, this orange network or the high attention network, is the set of functional connections that's stronger in people who are performing well on the task. And then these blue connections here is the set of connections that are stronger in people who are performing poorly. And so I want to emphasize that everybody expresses both of these networks. You just express them to a different degree. And it's not the case that all of these functional connections or correlation coefficients are positive and all of these are negative. It's simply that they're positively and negatively related to behavior. And so the next step in the approach is to formally relate strength in these networks to behavior, and we do that with a pretty simple linear model. So we simply look at overall how much each participant expresses the network by summing the functional connection strengths in the network and then relating that to behavior. And so you can see we get the expected effect such that people who express this network more strongly overall perform better on the task. And when we look in the low attention network, we see the opposite pattern, so people who express this network more strongly perform worse. And this is guaranteed by our analysis procedure, right? So this is, we've selected connections related to behavior, and then we see overall they're related to behavior. But you might be wondering, we've performed over 35,000 correlations. We may have overfit our model. How do we know that these features are actually predicting attention and they're not simply, related to our measure by chance? In order to do that, we now bring in data from the left out person. And we say, okay, how strongly did they express these networks? And we use that measure to predict how well we think they performed on this task. And so if we iterate through all of our subjects, leaving each one out once, we can get a predicted measure of performance for each individual. And so that's what I'm showing you here. On the x-axis is how people actually performed on the task, and on the y-axis is how we guess they performed based on their connectivity patterns. And when we make predictions using the high attention network model, we capture significant variance in performance. So we're explaining over 70% of the variance and how people are doing on the task just based on the brain data. We get similar performance when we make predictions with the low attention network model and a model that takes into account strength in both networks, which is interesting because it suggests that maybe they provide some degree of redundant information, and that's something we're exploring now. But I propose that we might be able to use functional MRI as a way to measure attention. So here you might be wondering, if I have to have you do the task in the scanner to predict how you're going to do, it's kind of an expensive way to measure performance, right? I could have just looked at the button press data. And so next, we applied our models to data collected while participants were not doing any task, while they were just resting in the scanner. And this answers two questions. So first, it tells us, do we need to have you perform an attention task in order to predict how well you pay attention? And it also asks whether the functional architecture of attention is reflected in the brain while you're simply lying in a scanner looking at a fixation cross on the screen. And so we can predict performance, so our predictions are not as good as they were when we used task data, but we are able to still explain significant variance in participants' performance just based on their functional connectivity patterns observed at rest. And one reason we think that the task predictions are so much better than the rest predictions is that when you're engaged in the attention task, it's perturbing circuits relevant to sustained attention and potentially magnifying these behaviorally relevant individual differences. And so my colleague Emily Finn has a nice review paper suggesting that we might think of psychological tasks like stress tests for certain processes. So an attention task might be analogous to a stress test for attention, for example. So we've identified a network that generalizes to predict performance on this continuous performance task. But are we really capturing something general about sustained attention, or are we just predicting how well people are going to perform on this idiosyncratic tasks with cities and mountains? To ask that question, we applied our model to data collected in pretty much as different a context as we could come up with. So we used publicly available data from children and adolescents with attention deficit hyperactivity disorder. And so in particular, we took advantage of the ADHD 200 data set, and we looked at data collected in China from 113 children and adolescents between the ages of 8 and 13. And so for each of these children, we had a measure of their resting state functional connectivity patterns, and we also had a measure of their ADHD symptoms. So these were rated by clinicians, and we had these scores for children that received ADHD diagnoses, but also for children who didn't have a diagnosis at all. So we're predicting this continuous measure of symptom severity in both patients and controls. And so the goal of this analysis was to apply our model that was trained in the Yale dataset to the current data to guess how we think each child or adolescent would perform if we were to hypothetically give them that same continuous performance task. And so here on the x-axis is ADHD symptom severity. So higher scores are more frequent or more severe symptoms. And on the y-axis is predicted task performance. And as you can see, we see the expected negative result, such that when we predict that a child or adolescent would perform well on the task, they actually show fewer symptoms or less severe symptoms of ADHD, suggesting that our model is capturing something in general about the ability to sustain attention, not just something specific about performance on one lab-based task. One thing you might be wondering is, are we just predicting the ability to, comply with instructions, be high functioning overall? And in order to ask whether our model was specifically predicting abilities related to attention, we asked whether predictions were related to ADHD scores, when controlling for IQ, and we found that they were. But the predictions were not related to IQ scores when controlling for ADHD scores. So the predictions are general in that they're generalizing across data sets, across measures of sustained attention, and across age groups. But they're specific in that they're predicting scores specifically related to attention. So I've talked a lot about these high and low attention networks, and you might be wondering what they look like. I'm not going to spend a whole lot of time on the network anatomy, but I just want to give you a sense of what these data-driven networks, where they are in the brain. So these are the high and low attention networks. Here, spheres represent brain regions or nodes, and they're sized according to the number of connections they have in both networks, and they're colored according to the network in which they have more connections. They might look like they take up the whole brain, but they don't. Together, they only represent about 4% of all possible functional connections in the networks. And so one feature of these images that I like to point out is that the colors are kind of washed out, but you might notice this big node here is gray, and that means that it has approximately equal number of connections in the network predicting better attention and the network predicting worse attention. And so what this emphasizes to me is that it's not, when we want to predict individual differences in attention, it's not the individual brain regions that matter per se, but rather statistical interactions between pairs of brain regions. So considering this functional connectivity measure is particularly important, at least in this context. Another way we can visualize the networks is on these circle plots. So here the semicircles represent hemispheres of the brain. So this is the right hemisphere, this is the left hemisphere, and then these colored lines represent sort of macro scale regions or lobes, and the gray lines represent functional connections between those lobes. There's a lot here. We could spend a whole talk on the anatomy of these networks, but I just like to make the point that when we use these data-driven techniques, they can sort of serve as hypothesis generators, so suggesting regions or connections that might be related to attention that we wouldn't previously have thought. But they can also sort of confirm previous hypotheses or agree with work. So one feature I think is particularly interesting is this many functional connections between hemispheres of the cerebellum in the low attention network. And this agrees with work in ADHD, for example, suggesting that cerebellar changes are particularly relevant. And so I hope I've convinced you that we can use brain-based models, and in this case, a connectome-based predictive model, to capture individual differences in sustained attention, and in particular, task performance in adults, and real-world ADHD symptoms in a developing population. But as I mentioned at the beginning of the talk, and as many of you may be experiencing right now, our attention changes over time, right? Like sometimes despite our best efforts, we're listening to a talk, we're trying to focus, but it just feels impossible. And if our model is really related to the ability to focus, we should find that it changes with changes in attention over time. And so now I want to talk about some newer work suggesting that these same connectome-based models are capturing changes in attention within a single person. These fluctuations in attention over numerous timescales have been emphasized with significant literature. So one way that we asked whether our model was sensitive to changes in attention over time was to measure the same person doing our attention task at many points in time. So we had a very nice volunteer agree to be in a study, and this person was scanned while they were performing the continuous performance task I gave you earlier. And then they were scanned again and again and again until they had been scanned 30 times. So we have, over the course of about 11 months, we have 30 fMRI sessions. And we have a functional connectivity matrix and task performance from each of those 30 sessions. And so now on the x-axis, I'm showing you that one person's task performance across all 30 of their sessions. And on the y-axis is how well we think they did based on connectivity in every session-specific pattern. And so again, these predictions are based in models defined in our 25 original adults, so totally independently defined model. And what we find is that the model is sensitive to this person's daily changes in task performance, right? So if the model was only sensitive to their average attentional ability, then we wouldn't expect to see any relationship between changes in this connectivity signature of attention and changes in behavior. But actually what we see is that the model is sensitive to, the person's best session and their worst session. Another feature of these data that I like to point out, this is only one subject, so I can't quantify it with statistics, but I just like to visualize it, which is if we look at this person's average task performance, it falls about here. And if we look at the average predicted task performance, it falls about here. So we're extremely accurate, not only in capturing changes in attention from session to session, but what appears to be sort of this person's overall, general, average, sustained attention ability. So we may have all experienced these sort of daily or moment-to-moment fluctuations in attention, but that's not the only way attention changes. So another way that attention changes is with pharmacological intervention. So I'm going to show you some data from a previously collected study where healthy adults were given either a single dose of methylphenidate or weren't given any drug at all. And so if you're not familiar, methylphenidate is a common ADHD treatment. It's better known by the trade names Ritalin and Concerta. And it's extremely effective. So we see symptom improvement in the majority of patients with ADHD, and we also see performance enhancements even in participants who aren't diagnosed with ADHD. And briefly, methylphenidate works by blocking dopamine and norepinephrine reuptake. And so what I'm showing you here is on the left here, high attention network strength in a group of adults given a single dose of methylphenidate before the scan, and a group of adults not given anything. And what we see is that, as expected, individuals given this attention-enhancing drug show this functional connectivity signature of better attention, so stronger high attention networks. And when we look in the low attention network, we see the exact opposite pattern. So it's not just that methylphenidate is having affecting functional connectivity overall, but it's selectively modulating these functional connections to give this person a functional connectivity signature of stronger attentional abilities. And these data were observed as people were performing a stop signal task, but we see the exact same pattern when these participants are simply resting. And so I think together, this set of studies suggests that the same models that predict individual differences in attention are also capturing fluctuations in attention within people and changes in attention due to a pharmacological intervention like Ritalin. And so before I move on to the last part of my talk, I just want to highlight the fact that we can use these functional connectivity patterns and this predictive modeling method not only to characterize individual differences in sustained attention, but individual differences in potentially an infinite number of abilities or behaviors or clinical symptoms that we might be interested in. So existing work from collaborators suggests that we can use these patterns to predict aspects of adults' working memory function, aspects of adults' fluid intelligence, autism symptoms in a developmental population, and work from many other groups is sharing and validating these functional connectivity biomarkers to ask to what degree do they generalize across data sets? Because if we really want to move towards an individualized, translational, precision neuroscience sort of applications, we need to confirm that our models generalize beyond the single data set on which we build them, and that they're robust to confounds like head motion, which I didn't talk about today, but is something we think about a lot, and I'm happy to answer any questions about. So in the last couple of minutes I just want to give you a little preview of some new work using a different brain measure to characterize a different important cognitive process. And so in particular, I want to talk about characterizing working memory in development with a different kind of brain-based predictive model. So one reason that I think it's particularly important to focus on these developmental periods, childhood and adolescence, is that we know that mental disorders, including ADHD, emerge and peak in adolescence. And so if we can predict symptoms and abilities in adulthood, that's great. But if we could predict these things earlier in development, we might be able to intervene earlier and afford people improved outcomes. And so I think one exciting opportunity we have is based on initiatives like the Adolescent Brain Cognitive Development Study that are collecting and sharing large developmental data sets. So every acronym here is a data set with MRI data from more than 500 participants in a developmental age range that's shared publicly. So there are resources available to train and test these predictive models in this age range. And what I'm going to talk about today is just one small example, but I encourage everyone to sort of make use of these resources that the NIH has invested heavily in, especially this ABCD study, which is what I'll be talking about today. So in this project, we use data from the first release of ABCD data. So this is data from over 5,000 9 and 10-year-olds collected at 21 sites across the United States. And we use their brain data to predict individual differences in working memory. And so why working memory? Working memory is a critical cognitive ability that allows us to store and manipulate information in mind. It's related to processing speed, fluid intelligence, and attention. And critically, working memory varies significantly between individuals and changes across development. Previous work suggests that these differences and these changes are supported by frontoparietal circuitry in the brain. And so here, what I'm going to show you is the relationship between task-based fMRI activity and individual differences in an out-of-scanner measure of working memory. So for each of these images, there's more than 2,000 individual participants that went into the analysis. And it differs slightly between the images just because of issues related to data quality and data exclusion. And so I'll be showing you images in which red or warmer colors show a positive relationship to working memory, and blue or cooler colors show a negative relationship to working memory. And so just to make this concrete, when we look at activation during a challenging working memory task, so a 2-back task, relative to a lower load working memory task, a 0-back task, we actually see that frontoparietal regions are significantly related to out-of-scanner working memory performance. And so what that means is that children who have sort of stronger working memory abilities activate these frontoparietal regions more in response to a working memory challenge than children with less strong abilities. And so this task-based activity signature seems to be differentiating these children. Interestingly, we don't see any differences if we look at activity in other contexts. So if we look at how strongly these children activate any region in the brain during an emotional versus a neutral block of this N-back task, we don't see any differences. Suggesting that it's not the case that if you have a stronger working memory, you simply engage frontoparietal circuitry more overall in any challenging context. Similarly, if we look at data collected during a stop signal task, an inhibitory control task, we don't see any working memory related differences. And again, we don't see any differences if we look in a monetary incentive delay task, a task related to reward processing. And so the reason I like to show all of these tasks is because it emphasizes that, psychological tasks are really a stress test for elucidating individual differences in brain activity related to behavior. So here, we see brain-related differences in children with stronger and weaker working memories when we give them an explicit working memory challenge, but we don't see those differences in other contexts.
Is this emotional?
Yeah, so this is an emotional N-back, meaning that some of the stimuli were emotional faces and some were neutral. Here we ignore that for the purposes of this analysis and simply contrast the 2-back relative to the 0-back blocks. But interestingly, this here is the exact same, it's the exact same data, but it's just characterized in a different way. Oh, this is very suspenseful. Here we go. No, it's fine. So this is data collected during the same task, but this is looking at high relative to low working memory load blocks, and this is looking at emotional versus neutral stimulus blocks. And so we only see the differences when we look at working memory related activity. Okay, so I hope I've convinced you today, or at least got you thinking about the idea that we can use these brain-based predictive models to predict individual differences in processes related to sustained attention and working memory. And we can also use them to characterize changes over time. And interestingly, it might be the case that the same models that predict differences between people are also capturing differences within a single person over multiple timescales. And finally, although I didn't dwell on this today, I think these kinds of data-driven predictive approaches can inform the functional architecture of attention and cognition in a way that serves as both a hypothesis generator and a way to provide novel evidence for previous findings. And so in general, I think these kinds of approaches, given that we're careful about testing model generalizability, controlling for confounds, and ensuring that our predictions are robust and meaningful. I think this can really help us move functional imaging from, as I said before, a science of group averages to a science of the individual. And so with that, I'd just like to thank my collaborators on the ABCD project and my grad school lab, my postdoc lab, and my brand new lab at Chicago, which is only a couple of months old. And thank you for your attention.
Thank you. Don't go away.
Oh. Thanks.
Are there questions? So I'm going to expect you people to talk, but while you're pondering your question, I have a question for you. So this is where I wasn't sure, because part of what you said, first of all, I thought it was a lot of fun, and one of the nice things is that you are working with normal people, healthy people, which is an everyday life. So I think that's really helpful in the context of this meeting. One of the things I was not sure about is how to put together the fact that you were able to pick up changes over time, changes with treatment, things like that, and yet you're saying that there is enough continuity that you can predict without the person doing the task. So I couldn't decide whether it's a trait, a state, or a combination of both.
That's a great question. And so I've been thinking about this a lot. To what degree do I think sustained attention is state-like versus trait-like? And what I think the analysis where I showed we could capture differences from day to day in a single person, but also capture their mean, I tend to think of it like we all have some kind of, just in a loose analogy, an attentional set point, which is on average how we're going to focus in a variety of contexts. But things like motivation, context, sleep, caffeine, all of these factors are influencing how whether we're achieving our maximum, whether we're at our worst, or whether we're around our average from day to day. So I tend to think about it like we're sort of fluctuating around our skeleton, and these models seem to be picking up on that kind of average skeleton mean ability, but also sensitive to changes. But I think one thing I didn't mention was, in addition to these sort of high throughput big data samples, more and more work is collecting lots of data on single individuals, and I think that will really help us tease apart these state-like versus trait-like effects.
So I'm going to push you. Do you think you can, or you'll get to the point where I could give you a whole bunch of these and you could sort one person out and say, this is her and this is him?
Oh, like use these patterns to identify people?
Yeah.
Yeah, so there has been work from Damien Fair's group and from Todd Constable's lab at Yale suggesting that you can use a person's connectivity pattern to pick them out of a group like a fingerprint. And there is work suggesting that this functional connectivity fingerprint, so to speak, is relatively stable across development. So you can even identify people years later. I think recent work has come out about that, but I'm less familiar with that work. But I think, right, so we do have these stable, quote unquote, trait-like connectivity patterns that predict overall abilities that don't change so much, so like fluid intelligence. But at the same time, we know that task states, cognitive states, pharmacological states are changing them to some degree.
So that raises ethical questions about having your brain signature online. But we can discuss that when that might be worse than your DNA signature. That's like much more evolved than just your DNA. So questions? Anybody? I know there's some smart people. Some of you I ate lunch with. So
it's okay if you want to admit that.
Okay, fine. Oh, okay, good. Smart people woke up. Please. We can handle more than one.
Hi, sorry, I just joined you. I'm Martha Tager with Bright Focus Foundation. So I just, my question is, does it, have you cross-correlated thinking types, more creativity, more, everyone laughs at the Myers-Briggs, but people who maybe have more, just a more tendency to integrate against attention?
So we haven't looked at the relationship between those factors and attention, but we have looked at the relationship between functional connectivity in general and personality traits and creativity. So I actually have a collaborative project that I think is a cool example of something I never would have done and they never would have done, or they were working on, but we sort of worked together. And they found that these connectivity patterns actually predicted people's divergent thinking abilities, and this generalized across multiple independent data sets collected across multiple continents. And then there's also work coming out from a number of labs suggesting that these same patterns predict aspects of personality. And so while we haven't sort of related those all to each other, I think these patterns are rich and they contain information beyond just how well you're focusing.
So, okay, this morning we learned that the circadian rhythm can have an impact on attention, reaction times, like with people being faster around 1 p.m., 2 p.m., maybe not today, but usually. So have you ever studied people during the course of a day?
That's a great question. No, but I've been really interested in this. So one thing I was worried about is like, what if we're just predicting what time of day we scanned people? We're not. So I looked at that scatter plot, and we had actually a pretty wide range, and our observed behavior and our predictions aren't related to time of day. However, I think it would be really interesting to look at data not just from a single person collected over 30 days, but collected over time in a single day to capture, when I say attention fluctuation, really what we think about are these sort of moment to moment, minute to minute, hour to hour changes. And so far I haven't looked at that, but I'm really interested in doing that.
Question. We know that in depressed individuals or lonely people, they tend to pay attention to the wrong things, like to be hypervigilant to social threats, to see more foes than friends. Have you studied the type of attention in your studies?
So I haven't looked at depression per se, but I think that's a really important point that I glossed over a bit in the talk, which is I'm talking about sustained attention, like it's always a good thing. But in fact, there are many different types of attention, and some of those are sort of an inverted U, right? So we want to be paying attention, but we don't want to be so locked in that we can't respond to external cues in our environment. And so I think characterizing brain signatures of different types of attention and then being careful to, not assume that always more is better will be important moving forward.
Yeah, and I think that is a great topic because it brings us back to the brain health topic. But thank you. We'll come back to it in the discussion.